Hello, everyone, and welcome to today's webinar, Developing Flow-Based Analytical Methods for Gene-Edited CAR-T Programs, How to Ensure an Efficient Transition from Early Research to Clinical Development. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. Let's get started. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Aaron Edwards, scientist, CAR-T in vitro biology lead, Beam Therapeutics. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Aaron, you may now begin your presentation.
Thank you. Thanks for having me. I'm going to start today's talk with an agenda of what I'll be speaking about today. First, I'm going to start with a review of CAR-T biology, the generations of CAR-T products, a reminder for those not as familiar with the stages of drug development. We'll then discuss some key reminders when you're building out CAR-T analytics infrastructures, 5 areas to really ensure an optimal transition of those facts-based analytics that you've created from research to clinical development, some areas for potential loss of data continuity and solutions, and then how we at Beam Therapeutics have integrated Miltenyi Biotech's infrastructure into our workflows from early research through clinical development. CAR-T, for those that aren't familiar, stands for chimeric antigen receptor T cells, which are a chimera of T cell receptors, which you can see in the figure, and the scFv region from an antibody that can be engineered into a T cell with specificity towards a cancer antigen or other antigen, depending on the area of interest that you're focused on. While CAR T cells are already complex products due to the nature of them being a cell therapy, each generation of successive CAR T products is becoming even more complex over time, with the integration of multiple signaling receptors, gene editing technology, and synthetic biology that is also requiring analytics to keep up with its complexity over time. These analytical assays also evolve and change many times throughout a product's development cycle. I'm showing a figure here with the trajectory from early research groups or development stages all the way through commercialization of your product. And with these analytical assays being developed, by the time you get to commercial, you might see something along the lines of the 7th version of such analytical assay being developed, where in the early stages of research, you then transition a first version of that assay to the next stage, receive feedback, fine-tune, improve, and so on over time. With the goal in mind to keep efficient transitions of these analytical assays throughout each phase of development, but also speed up the time from a concept in the early research stage to commercial, it's really important to minimize the changes made over assays so that you can have efficient translation of data, translatability, and speed going from early research through your clinical development. It's also important to keep in mind that every stage of CAR-T development and data collection are really opportunities to streamline these assays. whether it's research, preclinical phase, or manufacturing, it's important to realize that each of those phases can also have internal groups or external groups, whether that's your contract manufacturing organization or a research organization, or a lot of companies also have academic collaborators that they work with. And while these are great places to have data collection, they're also potential places for data variability. And it's important to keep that in mind as you go throughout the development process. And to highlight one specific area of this drug development process, just CAR-T manufacturing is multifaceted in itself and utilizes FACS analytics at multiple stages throughout that manufacturing process. And if you haven't gotten my point yet, I'm trying to make, these flow cytometry assays that you develop are used frequently and consistently from research through development, through clinical and commercial. And it's really important to know best ways to optimize and utilize these assays so that your data can be continuous and translatable from each phase of development. All steps of flow cytometry assays can also be sources of human error and variability, whether it's the cell labeling process, where you need to keep in mind lot-to-lot reagent variability, or even pipetting error, running samples on the cytometer, where you need to remember machine operations can change over time. How you set up that machine is critical. If you have different machines at different sites or different groups within a company, you need to keep that in mind and how they run across machines, and also operator variability. And then the final step, which is of course analyzing that data, population gating is something to keep in mind. That can be quite subjective from group to group, depending on how you gate. And the important question as you look throughout these steps of flow cytometry is, what ways can we identify these sources of variability early on in the process and try to prevent or reduce them accordingly? So I'm going to dive into some key areas that I think that we can make significant improvements to these sources so that you can ensure data reproducibility and translatability over time, with the end goal of hopefully speeding up your drug development process. For those of you that aren't as familiar with CAR-T analytics in particular, these flow-based assays for various stages can actually frequently overlap. So I've listed here on the left some various panels or assays that are frequently run within CAR-T analytics research or preclinical groups, whether it's viability, potency, phenotype, activation, editing for gene-edited products, phosphorylation, depending on your technology. And as you transition from this research and preclinical phase to a more clinical-focused development phase, those same assays are actually can be given a different name and serve a clinical or regulatory purpose for the viability or identity, purity, and safety. And I think it's important to realize that and recognize this overlap early on in the development process so that you can design panels that can be used from research through clinical. Before I dive into 5 areas that we can hone and optimize your flow cytometry-based assays, I want to list 3 points to keep in mind when you're creating a robust and sustainable analytics infrastructure. And number 1 is that humans make mistakes, and that is okay. Identify those areas where you think that there might be significant sources of human error and try to improve it as early as possible. Number 2, always have the next development stage in mind when making your analytics decisions, whether it's buying hardware, designing panels, or creating SOPs. You want to make sure that those are standardized from start to finish to ensure an optimal transition. And simplicity is key. While these products are extremely complex in nature and our analytics need to be equally robust to keep up with these complex products, the simpler that you can make your panels, the better, or simpler the operation of those panels, the better. I'm now going to dive into 5 areas that I think you need to focus on when really streamlining and optimizing your flow cytometry assays to ensure this efficient transition of assays over time. Number 1 will be the assay and panel design. Number 2 is the hardware selection and the design of that hardware. Number 3 is maintaining performance of your machines over time. 4, device-device synchronization and how to do that. And 5 is maintaining operations between groups and sites, which is key. But the first area in flow cytometry to keep in mind to streamline and optimize is assay and panel design, some considerations and some solutions. Design panels with as minimal spectral spillover as possible. And if you need to have panels that are complex and do have spillover into other channels, you'll of course need to compensate. And think early within that design of how you're going to compensate. And if needed, make sure that you standardize the methodology of that compensation across groups and sites. And if possible, and the hardware allows it, automate that compensation. You want to design these panels or assays for longevity across phases of development so that you don't have to change, retune, pause, and refocus every time another group or another stage needs to take over that assay. Think about if your panels are going to be compatible across multiple instruments. It's very common for groups or institutions to have particular favorites when it comes to hardware or software. And you really want to make sure that those panels are going to be compatible across stages and sites. And of course, controls. When possible, use commercially available controls. But if you need to, create a master cell bank. And if you have identified that a master cell bank is needed, start as early as possible so that you can validate and have the documentation necessary so that that master cell bank can be used for multiple stages. On some different solutions for these considerations, as I said, if compens is needed, standardize and automate when possible. Try to select reagents from a single company as well. I can't tell you how many times I've had panels where 1 or 2 antibodies are from one company and then other antibodies are from another. And if one company has a manufacturing issue with one of their antibodies, then your whole panel can't be used. And also ensure, this might sound very rudimentary, but really ensure that your assays and panels are robust before transferring to another group. It might work for one particular purpose, but try to really push the limits of those assays to make sure that you're going to be able to troubleshoot things that might come along in later stages beyond the optimal conditions for your product or your process. The second area to optimize your flow cytometry assays and processes is really the hardware selection and design step. So really keep in mind the design of your instruments. That not all cytometers are made the same way. They might have different optical components or detection components that can really change how your data is acquired or the ability to run your panels on different instruments. The display of those fluorescent values can also change from machine to machine. And not only machine to machine, but software to software. So try to be as consistent as possible, whether you're an early company able to standardize the hardware or software across groups and manufacturing sites, or if you're a mid-size or a larger company and you're able to alter what groups are doing over time, try to be as consistent as possible. Keep reproducibility in mind, whether it's running assays on the same instrument over time, whether it's running the same assay across the same model of instrument from either group to group or site to site, or vendor to vendor, for those of you that utilize external manufacturing organizations. keep reproducibility in mind, and hardware selection is an important stage to utilize and optimize keeping reproducibility in mind. Some solutions, if possible, use that same machine at every location that data is being generated. And if not, at least align on those optical and detection components. And for those, when you have identified that there are large differences in the hardware or software being used to analyze your fax analyses, or excuse me, your fax data, just have early conversations comparing those machines or softwares as soon as possible. The third area to ensure and optimize your flow cytometry assays is to maintain equivalent performance of your machines over time. So keep in mind what your control method is going to be. Are you going to use master cell banks? Are you going to use single stain beads or rainbow beads to then check those target MFIs that have been set and standardized and adjusted accordingly? Are you going to use commercial reagents? Another thing to keep in mind, it might sound rudimentary, but make sure that you're doing regular maintenance on your machines. It's extremely important to utilize those manufacturers' recommendations to not only do your daily, weekly, and monthly maintenance internal to a site, but also make sure the vendor of that machine is coming in to do a more extended calibration over time. Speaking of calibration, this is the first time I will plug in Beam, and Beam's use of some of Miltenyi's reagents and infrastructure is the ability to use their own calibration and, in combination, automated calibration on their max quant systems. So I have a picture below showing how easy it is to be able to take their calibration beads, whether it's daily or every other day, depending on what you've decided is most optimal for your assays. You scan a barcode, run the calibration, and instead of having to adjust everything manually, the system actually automatically adjusts for you. And I can't tell you, for those that have had to run calibrations on flow cytometers manually before, having a machine do it for you is definitely a breath of fresh air. So if you haven't seen how this is used, or some of how Miltenyi's instruments can utilize such automated calibration mechanics, definitely check them out on their website. The next area for optimization is really multi-site harmonization of those instrument settings and calibration and your SOP calibrations. Remember that each of those instruments have intrinsic differences with detectors and channels that can also perform with variability, whether it's the exact same machine that you have at multiple sites or different machines. They're all going to have their own variability from machine 1 to machine 2. Reagent and staining synchronization are critical. Keeping in mind lot-to-lot consistency, and also how you're staining your cells. One of the most significant areas that I've seen over time of data variability actually comes from how the exact same protocol has been run from group to group or site to site. And lastly, post-optimization of both your instrument and reagent and staining synchronizations, the ability to monitor your data over time and be able to either manually or hopefully automatically, or use an automated system to adapt when possible. Some solutions to these considerations is really make sure that you're setting up SOPs as soon as possible in your process. If you have standardized practices in place, it's going to pay dividends for you down the road, especially for those early research groups that are transitioning assays to later stage development groups. When possible, use pre-mixed antibody cocktails. Another place where I'll plug Miltenyi's infrastructure is in here. If you have been purchasing antibodies from one company, such as Miltenyi, over time, they actually provide a service where they can take your panels that you've been running and provide you with a pre-mixed antibody cocktail, so that either you or your external manufacturers don't have to individually add each antibody to your mix. You take your pre-set aliquot out, and you can remove that potential source of variability from that staining step. And as I said, using automated solutions when possible, and I'll actually go into that in a bit on how Miltenyi has their own solution to be able to use automated software to then calibrate instruments across multiple sites. And now I'll actually talk a little bit more about some of Miltenyi's reagents and software components that Beam has really been a fan of from day one. The first one is actually their Reaffinity antibodies that can be used as a solution that we do believe improve data and streamline your workflows. And I couldn't have made these images better, so I've taken them straight from Miltenyi's brochure. They are commonly generated with the key in mind to really have consistency and reproducibility from lot to lot. You see noted here that they have the mutated FC region, really keeping in mind eliminating that background that a lot of us are very familiar with when staining our cells. Also, they all have one universal IgG1 isotype. For those that have had panel design experience in the past, you can see a great schematic on the right highlighting that benefit of only having one isotype control needed. If you have a panel of, say, 10 different antibodies here that have multiple different isotypes, it is extremely useful to be able to transition those to Miltenyi's RIA system, to be able to only have one isotype control in all of your panels. We also stand behind their advertised benefits of being superior to hybridoma monoclonals in several key aspects, whether it's reducing that background, which I mentioned, by having them reach an FC receptor. You can see some of their own data that's generated here with a mouse monoclonal run on the left, or one of the RIA antibodies run on the right. With and without FC block, you can see that there is no change in that background signal with the RIA antibodies, but there is benefit from that FC block when using a mouse monoclonal, because you did need to block the background signal from that. Superior purity, it's quite common with hybridoma models, hybridoma monoclonal antibody production, to have contaminating light chains in your production. And you can see mass spec images on the right showing that contamination with the red asterisk compared to RIA antibodies, which are recombinantly generated, where you don't have that issue. And also enhanced specificity, showing the comparison between monoclonals and that RIA antibody from left to right. Another key piece of software that Miltenyi has that Beam is big fans of is their SmartGain technology, which actually allows for reduced time and effort to harmonize multiple instruments, whether it's within your lab, within groups, or within manufacturing sites across the world. Now on the left, I've listed traditional methods that usually you have to take to synchronize multiple instruments, whether it's starting out with identifying your master settings, validating those settings on your master instrument panel. You would then have to optimize your instrument settings on additional instruments to deliver that equivalent MFI from instrument to instrument. You would then monitor your gains in performance over time and really ensure harmonization from instrument to instrument and site to site. Miltenyi's SmartGain technology actually allows you to skip some of those, or have confidence in some of your final stages and really do this efficiently. So you would only need to identify settings and validate your panel on one instrument. You take all of those settings that you set up and validated from one instrument and take that to any other instrument with a simple USB load, and you can ensure that that same panel will run the same way on the same instrument with reproducibility and confidence. We've really had great success with this with multiple instruments on site and even tech transfers from site to site. So we're big fans of this technology. The last key consideration to keep in mind when optimizing your flow cytometry workflows is managing operations across groups and sites. So as I've been saying before, operator to operator variability is reality that's going to happen, whether it's the knowledge transfer or a tech transfer from group to group or site to site, whether it's day-to-day assay setup, calibration, compensation, and data analysis. The variability is going to happen, and you need to brainstorm ways to minimize that variability as much as possible. The final solution really is to also think about how you can simplify all of these steps and automate them as much as possible. The final place where I'll put a plug-in for Multini and their software and infrastructure is their express modes, where you can actually have a complete system to automate your sample run all the way through analysis. And they even have express modes tailored for CAR T cells in general, where you would start your run, walk away, the express mode would do everything in the analysis, and when you come back, your analysis report is ready to go. We actually think this would be a great utility for processes that you're really running in high numbers, such as autologous therapies, where you're running the same process over and over and over again, and you really want to remove that variability from your analyses. You can go on their site and check out, I believe they have about 12 different express modes specifically tailored for CAR T cell technologies. If you don't find one that is suited for you or your needs, you can actually work with them to create custom express modes that utilize your own data to generate an algorithm to do all of your gating, analysis, and report generation for you in a very efficient and streamlined manner. So now that you've kept those 5 key considerations to really optimize and streamline your flow cytometry workflows, what is the final ingredient needed to reduce the bench to bedside time? In my vote, I would say it's communication. Communication at every stage, as frequent and early as possible. Some key points of discussion to keep in mind during these conversations is asking yourself, can your early R&D panels or assays work for your later stage needs? So that would require communication from early research groups to some of your later stage analytical teams, and having a discussion about how you can design those panels very thoughtfully to not have to retool and retune at every stage of the process. If changes are needed, try to come up with a plan ahead of time. Really be proactive about being able to make changes to those processes and know how you're going to make those changes. Keep in mind when you are making changes to your panels, it's okay, but try to do so in a way that you can ensure data continuity, whether that's changing some fluorophores in the panel or completely changing hardware. You want to try to make sure that the data you've already generated in your development process can be used for later stages if you need to go back and reference that older data. Think about how to communicate issues. Issues will come up with your assays over time. Whether you have a manufacturing run that really highlights maybe a potential design issue. Be encouraging of communication of issues. And the last thing to keep in mind is adapting to new regulatory requirements. If you're at a company that has already submitted things to your regulatory agency, don't assume that that those same requirements are going to be exactly tailored for your next product. As we are building these complex therapies and adding more and more technologies to it, we need to be ready to adapt for those new regulatory requirements. And to conclude my talk today, I'll leave you with 5 things to keep in mind. These CAR-T therapies are going to require appropriate analytics as these products become more complex over time. So utilize those 5 key considerations that I listed earlier to ensure that your flow cytometry data can be optimized as early as possible and streamlined from early research through clinical development. Think early about how to build this infrastructure of reagents, hardware, and software that can be maintained throughout each stage, whether that's one brand, one company, etc. As I said earlier, early and frequent communication from these early research groups all the way through clinical development will be crucial to efficiently transfer those assays and ensure data robustness. And approach regulatory agencies as soon as possible to make sure what you're developing at those early research or preclinical stages really meets the needs of your product and development. And now I'd be happy to take any questions from the audience.
Thank you, Aaron, for your informative presentation. We'll now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, how do you implement standardized analytical readouts across the CAR-T manufacturing workflow?
That's a great question. I think this really requires a conversation from every group that's involved in the development workflow. So if you're in an early research group, bring in your analytical teams to see if there's a way to choose one brand or company or infrastructure that works for you in later stage development needs. If you know that you're also going to be seeking external manufacturing organizations to manufacture your product, have conversations early with them as well to see what hardware and software they have on site. And if possible, if there's room in the budget to be able to synchronize across sites, I think that is always great to keep in mind.
Okay, great. Thank you, Aaron. Our next question, if there are changes needed later on in an established SOP, how would you go about exacting those changes, and why is it easier to make adjustments earlier on compared to later?
That's also a great question. I would say, as I said earlier in the talk, you need to expect this to happen. You're going to make changes and establish SOPs, and hopefully you have a plan about how you make those changes. So we have done this on recently as well, where we've identified an area where we need to make a change in specific protocol. And again, it's communication, that final ingredient you really need to make sure you're doing things efficiently, but also that you're able to pivot efficiently. So identify the change that you need to make, bring all the groups together where that change will be impacting them. Also see if that change is going to impact any of your previous data. So if you're changing a panel, et cetera, pilot that change and see if you can get the same data that you've already generated in the past, or if the change was required to improve your data, really confirm that that change is necessary. To the second part of the question, I think it's much easier to make changes early compared to later due to some of the kind of manufacturing and regulatory requirements of later stage development. So in earlier research groups tend to have a lot of ability to pivot and change, say, panels, et cetera. But as you solidify and lock these SOPs in place, it's extremely challenging to start generating really extensive manufacturing run data, make changes, and be able to use those previous runs for, say, regulatory filings or important decisions that are going to have an impact on your product development long term. So I think that if you remember anything from this answer, identify the need to make changes as fast as possible and try to do it early on so that you don't have to make significant changes later on in the process that might be very costly for you and your company.
Okay, great. Our next question: when dealing with multi-center sites, how does one go about ensuring efficient tech transfer, and what are some hurdles to potentially prepare for?
That's a great question. Ensuring efficient tech transfers, number one is to have a solidified and robust SOP in place. Look at your SOP, make sure it's as detailed as possible so that when you're communicating or showing another site how to do either the staining or running, there are no questions that could be asked that aren't listed. Be clear and communicative. Traditionally, people have been able to travel from site to site to perform a tech transfer, and these days, especially in the time of COVID, it's not always possible to make that trip. Something that Beam has actually used recently is our webcams, where we've actually recorded one site doing a certain assay and the other site doing the same assay, so that we can be a little bit flexible given some of the safety requirements these days. Some hurdles to potentially prepare for. Even though you might think you have every detail possible listed in your SOP, something will come up where an operator at one site has been performing an assay one way and another was performing it slightly different. That might have been skewing your data. That's okay, be ready for it, and just make sure that it's communicated in the next version of your SOP. Another place where I'll plug in Miltenyi and their technologies is being able to pick systems that have things like Miltenyi Smart Game technology has actually been a lifesaver for Beam in a lot of ways, where we can have all of our instrument settings done at our master site, in those settings to another site, and really have confidence that the data on one machine was being run, ensure confidence that the data on one machine will be the same data on another site. We've had really successful tech transfers using some of Miltenyi's machines recently, and we're very thankful for that. Another hurdle to prepare for is being able to just make changes to your SOPs. Not only are you going to find that you didn't have details listed that's causing some operator-to-operator variability, but you'll need to make changes. And depending on the site and their ability and flexibility to make changes, it can be more or less effort. But be prepared for that. Try to be flexible and be as communicative as possible.
Okay, great. We have another question here. In addition to having instrument redundancy to address consistency, are there supplemental tools or protocols that can ensure instrument and data harmonization? If so, would those tools be different in a research versus industrial space?
That's a great question. I don't think every institution, of course, has the ability to have instrument redundancy. I mean, for those of you who aren't familiar, if one instrument shuts down, you have the ability to use another instrument. I think that the only way, if you don't have the ability to have multiple instruments on site, is to make sure that you're maintaining the one you have to the best of your abilities. There are best practices listed on every manufacturer. that really will allow you to make sure that your machines are running efficiently and smoothly over time, because all of us in the flow cytometry space know how much of a pain it is when you have a laser that isn't aligned properly, and you have to have a technician come on site that really to fix your machine, which can really hinder your ability to acquire a lot of your data. I think it does, going to the second part of the question, the tools can be a little bit different for the research versus industrial space, but they don't have to be. I think one of the reasons that we've been big fans in Miltenny is being able to have that same instrument from early research groups throughout later research and development groups. One of the cool things about their tech is that they have live support, where instead of needing to schedule and shut down a machine for multiple days, it's possible to have live support with them to be able to fix your machine right there and then on the spot by syncing into your device. I think the biggest differences that you might see between that research and industrial space is potentially the ability to have two instruments, but I don't see why some of those tools or protocols need to be that different.
Okay, thank you, Aaron. We have time for one more question. What are the hallmarks of variable data, and how does a company address this? What are the consequences of unreliable data in the CAR-T manufacturing space?
Yes. For flow cytometry, I think one of the biggest spaces where you'll see your data be variable can be, MFI of a target. When you get really comfortable with different protocols, a lot of that variability might come visually, where people that have run panels over and over and over again might see something shift, especially in one of your controls. If you say have a master cell bank that you've been running, you really make sure that those controls, or however you're harmonizing or synchronizing your performance of your machine, make sure that those controls are running the same way over time. I think it's also possible that one of your lasers has shifted slowly over time, which is going to happen. And I think to the trained eye, you can see it visually, but there are also ways to do that quantitatively, depending on how your institution has set up some of those QC parameters. So, have a discussion with your group early on about how you want to address this, because you might have different procedures in place, but definitely be prepared for it. Some of the consequences for some unreliable data, if you've, as I was saying earlier, if you've gone through multiple extremely expensive manufacturing runs and then realize that one of your panels actually is displaying data that isn't actually of the nature you thought it was, depending on where in that manufacturing process you are, it can be extremely challenging if you're planning to use one of your runs, say, for regulatory filing. It's not the end of the world, but it is possible that you might have to either redo the entire run, or hopefully, best case scenario when this does happen is that you can take retains from that run and utilize a new panel to be able to reacquire those analytics on that manufacturing run.
Great. Thank you again, Aaron. Thank you for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Miltenny Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.